MSK, anatomy and physiology, muscles. All right, so muscles are relatively straightforward. We know kind of how the whole entire process works if you remember it from M1 year, but I'm going to run it through just really quick one time how the whole entire process is going to work out. There's two different types of muscles that we're going to discuss. First, we're probably going to, honestly, I'll let you know when we're discussing different types of muscles, but once again, it's not too important, the differentiation. What I will say, though, is I will pause and let you know when I remember getting certain questions, because this is just a lot of information that's not too high yield because it's just physiology, and I'll let you know what we really should know for question stems. So what happens first is that we have an action potential. All right, so action potential, and this is striated muscle, so all striated muscle. So action potential is going to open up presynaptic voltage-gated calcium channels. Okay, so action potential is going to come, open up calcium channels, and it's going to cause acetylcholine to be released. Right, so this is at your presynaptic cleft. Right, acetylcholine release is going to go. It's going to bind to the postsynaptic acetylcholine receptors, and it's going to cause muscle cell depolarization. All right, so this is known as the motor end plate, and acetylcholine is going to bind there, and it's going to cause depolarization. That depolarization is going to travel, okay, over the whole entire muscle cell, and it's going to go into T tubules, okay, which are those burrows within the muscle, because you want the muscle to contract at the same time. So we have these T tubules that kind of go through the muscle, deep into the muscle. So the depolarization is going to travel throughout the muscle, down T tubules, and it's going to cause membrane depolarization. And what happens after membrane depolarization is that you're going to have voltage-sensitive DHPR, dihydropyridine receptors, that are going to be opened. And they're going to mechanically open ryanodine receptors, RRs. Okay? When they open ryanodine receptors, that essentially is going to cause calcium to be released from the sarcoplasmic reticulum of the muscle. Now that calcium is free from the sarcoplasmic reticulum of the muscle, what it's going to be able to do is it's going to be able to travel and cause contraction. And how does it cause contraction? Calcium is going to be able to bind troponin, okay? And troponin is going to pull tropomyosin off of the active sites, okay? So tropomyosin is what's blocking the myosin binding sites on the actin filaments. So calcium is going to bind troponin C, which is going to help shift tropomyosin to expose the myosin binding sites. Now that those sites are exposed, myosin heads are going to be able to bind to actin, all right, and then cause essentially muscle contraction. Now, how does muscle contraction occur? Well, muscle contraction is going to occur via, one, the release of a free phosphate. When you release the free phosphate, it initiates a power stroke, okay? Then, during the power stroke, myosin is going to pull on the thin filaments of actin, okay, and it's going to shorten the muscle. And specifically at the sarcomere level, the two things that shorten are the H and I bands. So remember, high goes by. So when you're looking at the sarcomere, it's like sarcomere from like a, I forget what they call it. I think it's like electron microscopy imaging. I don't know, where you see like the black and gray image of a sarcomere. You have to be able to notify where the H band is and the I band is, because that's going to shrink during contraction. So high goes by. All right. Now, the A band remains the same length, obviously, because that's just our myosin, all right? So keep that in mind. Now, ADP is going to be released at the end of the power stroke, and then binding of a new ATP molecule is going to cause detachment of the myosin head from actin. So you have to have ATP in order to detach the myosin head from actin. And then calcium can go get placed back into the sarcoplasmic reticulum, and then kind of everything causes relaxation. So ATP hydrolyzes into ADP and free phosphate, which results in myosin head returning to a high energy position, which is known as the cocked position. Okay? Now, the myosin head can bind to a new active site on actin, right, to form a cross bridge only if calcium is still present there, right? Because if calcium goes back and is sequestered into the sarcoplasmic reticulum, then you no longer have, essentially tropomyosin is going to cover our actin, our binding sites. Okay, so reuptake of calcium by the sarcoplasmic reticulum is going to be done via the sarcoplasmic or sarcoendoplasmic reticulum calcium ATPase channel, which is known as SERCA, which leads to muscle relaxation. All right, so that whole process, I just said it just so that you can fully understand it, what happens. But the key takeaway points, the first takeaway point is during muscle contraction, what initiates it is calcium and troponin binding in order to pull off tropomyosin. Okay, so calcium plus troponin pull off tropomyosin in order to expose the active binding sites. Okay? Number two takeaway point is that you need ATP in order to detach the binding sites. Without ATP, the muscle stays contracted. And this is kind of the pathophysiology behind rigor mortis. When a patient passes away, they have essentially all of their muscles contracted still. The reason behind it is because there's no more ATP. So all those muscles stay contracted because ATP no longer is present to detach the active site. All right? Lastly, you need to know where the Z line is on like the sarcoplasm. Like if you were to take a look at the sarcomere on imaging, know where the Z line is, know where the H band is and the I band, as well as the A band. Know that the H and I bands are going to shrink during contraction. Know that the Z line is present the whole entire time, and know that it's like the darkest portion. Okay, be able to identify it. All right, so overall, that is our motor neuron action potential that travels through the muscle and causes muscle contraction. Now, there's two types of skeletal muscle fibers that you need to be aware of. You have type 1 and type 2. Type 1 is going to be slow contractions. It's going to be red fibers, and it's usually going to be predominant of oxidative phosphorylation, okay? And there's going to be tons and tons and tons of mitochondria here. The reason behind this is for muscles of posture, right? So muscles that are going to be continuously contracting throughout the whole entire day, right? So we need these slow contracting muscles with a bunch of mitochondria, which gives it the red color, right? That are undergoing oxidative phosphorylation so that we can have sustained contraction for those like postural muscles and endurance training muscles, right? Makes sense. Now, on the other hand, we have type 2. These are our fast contracting muscles. They appear white because there's a decreased amount of mitochondria in it. The reason behind that is because it undergoes anaerobic glycolysis. Well, this is for our quick used muscles like sprinting and weight training. So we need ATP and energy quickly. So that's why it undergoes anaerobic glycolysis rather than oxidative phosphorylation. Okay. But we don't need it for long periods of time. So that's what makes anaerobic glycolysis very useful in type 2, because we need it for a short period amount of time, but it's just like a quick and like active type of time period. Okay. Now, skeletal muscle adaptations, we talked about atrophy versus hypertrophy. We know that, right? So in hypertrophy, you're going to see myofibrils increased as well as myonuclei increased. Compared to atrophy, you'll see myofibrils decreased and myonuclei decreased, okay? Now, for vascular smooth muscle contraction and relaxation, this is going to be done by nitric oxide. So this is our smooth muscle contraction and relaxation. So it's not going to be done by the action potential that we discussed previously. That's striated muscle. So for smooth muscle, what happens, and essentially what you need to know is contraction-wise, the calcium is going to bind calmodulin complex, right? Rather than our troponin, where calcium binds troponin in striated muscle, calcium in smooth muscle is going to bind calmodulin, and that's going to activate myosin light chain kinase. Myosin light chain kinase activation is going to cause contraction, okay? Now, opposite occurring, nitric oxide is going to activate CGMP, which activates myosin light chain phosphatase, and that leads to relaxation. All right, so keep those two things in mind. It's very straightforward for vascular smooth muscle contraction and relaxation. Lastly, we have our muscle proprioceptors. The key takeaways here, there's two things: muscle stretch receptors and Golgi tendon organs. These are just specialized sensory receptors in our muscle that help us protect our muscles, okay? So we don't go too crazy. The first one, the muscle stretch receptors, they monitor and maintain muscle length, okay? So they're going to be able to sense muscle stretch. If the length becomes too long or if the stretch becomes too tough, then muscle stretch receptors are going to be activated and stop the body from doing anything. Golgi tendon organ is going to be monitoring and maintaining muscle force, and they sense muscle tension. Okay, so those are the key takeaways. Muscle stretch receptors monitor and maintain muscle length by sensing the muscle stretch. Golgi tendon organ is going to monitor and maintain muscle force by essentially sensing muscle tension, okay? That's the two key takeaways from each of the muscle stretch receptors and Golgi tendon organs. So that's pretty much overall the muscles. Not too high yield. Once again, just remember the things that we discussed and you should be fine.